Assalomu alaykum hurmatli tinglovchilar. Biz Buxoro davlat tibbiyot instituti mikrobiologiya, immunologiya va virusologiya kafedrasining dotsenti PhD Mustafaeva Feruza bo'limdaman. Bugungi ma'ruzamiz ya'ni ingliz tilida bo'lib, bunda ingliz tilidagi ma'ruzamiz mikroorganizmlarning morfologiyasi, strukturasi, bakteriyalarning strukturasi va o'rganish usullari bo'lib hisoblanadi. Shu haqida biz inglizcha sizga ma'ruzani o'tib bermoqchimiz. Microorganism morphology, ultrastructure of the bacteria, the factors and research methods. Today we consider about these, and if you look at these contents, we must see about the introduction, morphology of the bacteria, shape, size, arrangement, ultrastructure of the bacteria, cell walls, and plasma membrane, capsule, pili, femora, mesosomes, cytoplasmic inclusions, flagella, endospores, nucleus, and plasmids. Introductions. Bacteria is uncellular free-living microscopic microorganisms capable of performing all of the essential functions of life. The process both deoxyribonucleic acid or DNA or ribonucleic acid RNA. Bacteria is prokaryote microorganisms. They do not contain chlorophylls. They occur in water, soil, air, food, and all of the natural environment. And they can survive extremes of temperature, pH, oxygen, and atmospheric pressures. Morphology of the bacteria. The morphology of the bacteria depends on the number of the factors, such as strains studied, the nature of the culture mediums, and the temperature and time of the incubation, the age of the culture, and number of the subculture it has undergone. Bacterial morphology is best studied by microscopy after the apparently by staining the bacteria. Microscopes helps in identify the following morphology characters of the bacteria. And shape, if you look at the shapes of the bacteria, we can see about the three types of the shapes. For example, in here we can see spherical shapes, rod-like shapes, and spiral shapes. When we wait under the light microscopes, most bacteria appear in a variation of the three major shapes. We say about the rod bacillus, the spherococcus, and the spiral types is the vibrio. And we can, in here we can see cocci, bacillus, and spirella. Now I told about that. And in here we can see shapes, many of the shapes, bacteria. For example, is a staphylococcus. If you look at under the staphylococcus, you can remember about the grapes. And in here staphylococcus aureus shapes. And in here we can see Mycobacterium tuberculosis is a bacillus shapes, and is a Vibrio cholera is a spiral shapes. And Streptococcus, if you look at about the Streptococcus, we remember about the neckler, woman neckler, they located like that. And Streptobacillus, bacillus of Anthractis, and Spirellus, Helicobacter pylorus. We know about the Helicobacter pylorus is today one of the widely spread in the world. And if you two of the coccus located two of them, we name about the Diplococcus. It's like that. If we see under the microscope like shaped bacteria, we can name name the Coccobacillus, and Spirochaetes bacteria. We can see one of the widely in the world Treponema pallidum. Size of the bacteria. The size of the bacteria is too important for us. For example, virus with kinds of the bacteria and also related to the age and external environmental. Size of the bacteria. In here we can see many types of the bacteria. Bacteria are too small to see through the naked eye. They does not necessarily mean that bacteria are of the uniform size and shape. In fact, bacteria come in a great many sizes and several shapes too. And in here we can see some of the bacteria. Some of the bacteria. In here we can see some bacteria. It's a look at is one of the macrophages, and bacteria types is coccus, bacillus, prolims, paracheta, bonding and appending bacteria, and filamentous bacteria too. Gigant bacteria is one of the gigant bacteria is Thymomericant magnificent, is the world's biggest single-cell bacteria. It's up to the 2 centimeter long and is visible to the naked eye. And we can see announce a little of these bacteria too today in the world. For example, Epilocycum phyllogense, another very large of prokaryote with a cells longer than 600 nanometers or 0.6 millimeters. This bacterium is phylogenetically related to the endospore forming bacterium. Clostridium is found in the gut of the surgeon fish. Smallest bacteria. Smallest bacteria is a mycoplasmas, about the same size as the largest viruses, poxviruses, and are the smallest organism capable of exciting outside a host. Okay, and the surface and volume ratio. The surface volume ratio is a spherical bacteria of 1 nanometer in a diameter is a high, is compared to spherical eukaryotic cell having a diameter of 20 nanometers. Arrangement. Bacteria may arrangement slightly in pairs, in tetrads, or in packets of 8. Alternatively, they may anchored in the chains short or long, as the case of cork. Bacillus may be arrangement and random in short or long chains, in the Chinese letter patterns, or in bundles too. Coccus. If you look at Diplococcus, is the cells divided into one plane and remain attachment predominantly in pairs. Pneumococci. Streptococcus cells is divided in one of the plane and remain attached to form chains. Streptococcus and Tetracoccus cells is divided to planes and forms groups of 4 cells. And Sarcinias cells divided is 3 planes and the regular pattern producing or capital arrangement, and Staphylococcus cells divided in 3 planes in an irregular pattern producing bunches of coccus. In here we can see Monococcus, Diplococcus, and Streptococcus, and division along the Tetracoccus, and for example Sarcinias and Staphylococcus. It's a very coccus shapes we can look in here. And arrangement coccus, in here you can see is a 2 coccus we name about the Diplococcus and Streptococcus, and is a we name about the 6 of them, for example Sarcinias and Staphylococcus, and if you can to see 4 coccus we name about the Tetracoccus. In here we can see under the microscope is the shape of this bacteria, how they look at under the microscope. In here we can see some nanometers and location of this bacteria. And it's co-arranged in the cultures. When we painting some complex painting, we can see some coloring this is bacteria too. And bacillus is a single diplobacillus in pairs, and streptobacillus in chains bacillum, subtilis, and trocheminus, palisadis, Chinese letters like, and other types of the bacteria. In here we can see monobacillus, and in here we can see 2 of the bacillus named about the diplobacillus, and chains of the bacillus are called streptobacillus, and palisadis rode side by side or in X, Y, or other figures. In here we can see some types of the figures of the bacillus. Okay, in here, for example, Coccobacillus shape is, remember our Coccobacillus and Bacillus and Palisadis and Diplobacillus. In here we can see 2 Bacillus and Streptobacillus. And here we can see under the microscope how to locate these bacillus and shapes, and we can see like that. And here we can see like that under the microscope of these shapes of these bacteria, bacillus shapes. Okay, spiral shaped bacteria we can name about spiralis and sprachetes and vibrios is the enter all of the three types of these bacteria. And in here we can see some sprachetes and helicoformes and other forms of the bacteria. Flagellum, the organ of vitality, one of the important organelles for the bacteria. We located in here so many types of the flagellas. Is a bacteria flagella hair-like and thin helical appendages. Is a man be about 12 or 13 nanometers and several times longer than bacterial cells, and most of the organisms that produce capsules are not motile, and none of the cocci of medical importance is motile. Arrangements of the bacterial flagellas we can see in a many types of the flagella located in bacteria. For example, in here we can see one of the flagellas we name about the monotrichus, and we can see in many of the flagella located one side of the bacteria we can name about the lophotrichus, and in here two sides of the bacteria located one flagella we name about the amphitrichus, and if you see under the microscope we name about the peritrichus. Peritrichus mean about around all of the bacteria located many flagellas. And here is you can see like the pictures of these, for example, how to locate flagella in bacteria. For example, monotrichus, amphitrichus, and cephalotrichus, and peritrichus, and lophotrichus of them. Structure of the bacteria. The flagella is hollow and rigid cylinder composites of the three parts, and based body, a hook, and filaments. And we know about the gram-positive. Bacteria world we divided about two big world about the gram-positive and gram-negative bacteria. Gram-positive bacteria organism has two basal body rings. One is a peptoglycan layer and one of the plasma membrane. If you look at gram-negative organism have four rings. It's the L ring associated with lipopolysaccharides, and P ring is associated with the peptoglycan layers, and M ring is embedded in the plasma membranes, and S ring is directly attached to the plasma membrane. In here we can see some gram-negative cell walls and gram-positive cell walls, and in here we can see some differences between them. For example, differences there is gram-negative is the peptoglycans, and in gram-positive we can see some peptoglycans too, and in the gram-negative have other membranes, but we cannot find the gram-positive bacteria, for example, as outer membranes in gram-positive bacteria. Function of the flagella, about one of the important, for example, function of this is a movement in the cells, and they act as sensory organs to detect temperature. And we can see method also to study bacterial morphology and structure, and staining methods. We know about the simple staining methods, differential staining methods, gram staining, acid-fat staining, and special staining methods, the spore staining methods, the flagella staining methods, and capsule staining methods, and negative stainings. Today we can see some variety types of staining in the world, and all of this important to us. And now I can say that we continue our lessons and other lectures. That's why I can say that thank you for attention. Our lecture is finished.